current students and 3.2 million alumni. IGNOU has also been accorded the highest grade of academic excellence, A++, by the National Assessment and Accreditation Council, or the NAC. Now, during your time at IGNOU as a learner, we will help you complete your program of study with world-class printed materials, audio and video programs. We have more than 35,000 academic counselors who will guide you till you complete your program of study. We also provide live interaction with your teachers at the headquarters via teleconferencing mode and live interactive radio counseling besides this, supported by regular TV and radio broadcasts. The School of Humanities was created in 1988, and we welcome you warmly to join any of our programs of study. IGNOU degrees, diplomas and certificates are recognized not only in the country but are also very well received across the globe. We look forward to your journey with us, and we welcome you once again. The School of Humanities of the Indira Gandhi National Open University offers more than 26 ODL programs, which is open and distance learning programs, and now more than 10 online programs. A very interesting feature that we have, a very interesting program that we have on offer right now is the BA English Honours. Now, in this particular program, what we've done is we've taken a large number of courses from English, which means is English as a discipline. And then we have also put in other interdisciplinary and skill-based elective courses. So all of them combine to give you the BA English Honours, which gives students the exposure to subjects which is related to English literature as well as other disciplines, and it will introduce you to various aspects of literature through the core, the discipline centric, the ability as well as the skill enhancement and generic courses. Now, it also exposes learners to the importance of interdisciplinary study, which is in keeping with the National Education Policy 2020. And how do we do this? Because we enable learners to opt for courses which are outside their discipline, not just from English. Now, the eligibility for this program, the BA English Honours, is you need to have a 10+2 grade. And for those students who do not have 5 core subjects, right? Especially if they have a SUPW course as the 5th subject, they need to pursue one senior secondary level course from the National Institute of Open Schooling, New Delhi, or any of the state open schools, so that you meet the minimum eligibility condition of having passed in minimum of 5 core subjects. The medium of instruction, needless to say, is English. The minimum duration of the program is 3 years, and the maximum your registration is valid is for a period of 6 years. Now, the BA English Honours program is offered both in the January as well as the July cycles of admissions. The fees for this particular program is Rs 4,200 per year. So in a total, you pay 12,600, and you also need to pay a registration fee of 300, which is only at the time of admissions. Now, in the BA English Honours program, let me tell you a little bit in detail. We have 14 core courses of 6 credits each. So your core courses will contribute to 84 credits. The list of courses is BEGC 101, 102, 3, 4, and it goes on till 114. And we begin with Indian classical literature, and we come down right up to post-colonial literature. Then we have the discipline-specific elective courses. So you need to opt for 4 of these courses, and these courses are of 6 credits each. So it's a total of 24 credits. Now, the discipline-specific elective courses are offered in the 5th and the 6th semesters of the BA English program, and you have understanding prose, understanding drama, understanding poetry, and understanding the novel. Then you have ability enhancement courses and skill enhancement courses, like I told you earlier. And these are, you have to do 4 courses in all, and each course is of 4 credits. So that means a total of 16 credits. The ability enhancement compulsory course is compulsory. One in the first year, you have to do it in the first semester, and the other in the second semester. So both the ability enhancement compulsory courses you will be doing in your first year of BA English Honours. Then you have the skill enhancement courses, or the SECs, which are basically value-based or skill-based courses, and they are designed for helping you develop competencies and skills in various areas. And these are on offer in the 3rd and the 4th semesters. So these are 2 courses, 4 credits each, so that means 8 credits, right? Then we have generic electives. Again, 4 courses, 6 credits each, 24 credits. Now, the generic elective courses are the other type of electives that are on offer in the first, second, third, and fourth semesters of the program. So, you're going to be doing it in the first year of your BA and the second year of your BA English honors. And these courses are interdisciplinary. Now, that in short was the BA English Honours program, and if you need further details, you can log on to the website www.ignou.ac.in, and from there you can also navigate your way to the School of Humanities and look under programs. Or you can also write to the program coordinator, School of Humanities. Her details are given below. Thank you.
Dear learner, welcome you all in the course of Immunology BBYET 141. Basically, it is a elective course for the BSc General. And dear learner, my name is Dr. Siaram. I am an Associate Professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, I want to discuss today very important topic that is known as the component of immune system. Dear learner, what is the expected outcome of today's session? So after studying of this unit, you will be able to define innate immunity. What is the innate immunity? Pattern recognitions and different type of pattern which are available on the host cell, pathogen which are associated, or you will be learn different type of the patterns which are available on the host cell. And after completion of this session, you will be able to explain the mechanism of phagocytosis using the flow chart. And after completion of this session, you will be able to explain inflammation and list its type, and you will be also able to describe the mechanism of the inflammation. So these are the expected outcome for today session. My session is divided into two parts. In part first, I will discuss in detail what is the different barrier of the innate immunity, like physical barrier and chemical barrier. And another thing, I want to discuss the natural killer cells, which are very important, which are found in our body. They will eliminate the infections. But in case of the part second, we will discuss the complement systems. It is another very important part of the component of the immune system. And phagocytosis, inflammations and their mechanism. So I want to discuss one by one In case of part second, it is the innate immunity. Innate immunity help the body recognize common pathogen molecule present on the surface of the pathogen. So, number of the things, specific molecule which are present on the surface of the pathogens, innate immunity will recognize it. It is the first non-specific line of defense of the bodies present from the birth, and different type of the barrier like anatomical barrier, inflammatory, physiological and phagocytic barriers which are available in the innate immunity. They will provide the immunity. Ultimately they are able to fight against the particular pathogens. I want to discuss one by one the different type of the barrier. First one is the physical barrier. Skin is very important part of this, and it is the largest body organs and that provide from the infection. It has an epidermis and dermis. The thin epidermis has keratin deposition and dead cells, while the thicker dermis has connective tissues, the blood vessels and sebaceous and sweat glands. And sebaceous maintain pH around 3 to 5. So you can see it. The sebaceous gland, they maintain the pH around 3.5, which inhibit the growth of microorganism. Microorganism will not survive at particular pH. And some microorganisms, they are able to survive, or they are able to cause the pathogenesis. Lysozyme is naturally occurring enzymes in the body, secrete such as tears, saliva and sweats, and they are able to kill the pathogen. Mucous membrane secrete the mucus that can protect from the microorganism by trapping. So these are the physical primary physical barriers. And another is physiological barrier. The different type of physiological barriers. It may be temperature, it may be pH, or it may be some soluble molecules which are present in our. They are able to maintain, or ultimately microbes will not grow. In case of temperature, the normal body temperature inhibit most pathogen. The example, chickens are anthrax resistant due to their high body temperatures. In pH, each microbes has optimum pH to grow, but stomach has 2.9 pH. They are able to eliminate the microorganism. They are able to kill the microorganism. Sebaceous gland, it maintain low pH and discourage the growth of microorganism. In soluble molecules, the body circulates some antimicrobial molecules, or ultimately it will provide. It is, you can say, it is a physiological barrier. They are able to remove the pathogens. They are able to reduce the pathogens. And it is a very important table related to the activity of the various antimicrobial agent. So these are the three molecule lysozyme, interferon, complement systems. They are able to kill the pathogen. They are able to kill the tumor cells. The lysozyme activity, what is the activity of the lysozyme? Found in tears, saliva and other mucous membrane secretion. It cleaves the peptidoglycan of the bacterial cell wall and thus they eliminate pathogen. Interferon create an antiviral state of the body, or ultimately the immune response will be generated after infection of the cell with particular viruses. And third one is the complement system. It is circulating inactive protein. It may be activated by formation of the antigen antibody complexes, by reaction between complement protein and cell wall complement of the pathogens. So these are the three very important molecules which are involved in the reduction of the pathogens. Another barrier that is known as the phagocytic or endocytic barriers. It is a next line of the defense against the pathogens. They have passed through the epithelial cell barriers. The phagocytes is a type of endocytosis carried out by macrophage, neutrophil, dendritic cells, and monocyte. So these are the four very important cells which are found in our body. They perform the phagocytosis, or ultimately they can engulf the pathogens. Endocytosis is the process in which the cell take in material from the outside of by engulfing and fusing them by the plasma membrane are killed by the phagocytosis. So these are the another very important barriers, phagocytic and endocytic barriers. And it is another barrier that is known as the inflammatory barrier. When the tissues damage occur as a result of the wounds or the pathogen invasion, inflammatory barrier contain the series of the events. This is triggered by the factors that are secreted by the damaged cells and they help to prevent infections. So it is very important. You can see in this figure, it is the tissues are there. Here is the injury. So ultimately, inflammation will take place, or they will not allow to move pathogen in the blood vessels. The anatomical and physiological barrier are physical barrier, while the phagocytic and inflammatory barrier are the chemical barriers. So these are the inflammatory barriers. They can provide the immunity against the pathogens. And another thing is pattern recognitions. The immune system have ability to recognize the molecule present on the cell surface of the microbes or tumors. An innate immune system has evolved a mechanism capable of recognizing this repeating pattern known as pattern recognition receptor. So you can see in this figure, these are the several receptors are there. They are able to recognize only particular protein that is known as the flagellin protein, which are available on the surface of the microorganism. They are able to recognize with the particular receptor that is known as the pattern recognition receptors. And it is a several family proteins. It express onto the many effector cells, especially on the professional APC, macrophage, dendritic cells, and B cells. And they are available into the transmembrane and cytosolic pattern recognition receptors. The pattern recognition receptor, it is divided into three classes: secreted pattern recognition receptor, endocytic pattern recognition receptor, and signaling pattern recognition receptor. Principal function of the PPRs is the opsonization. It is a very important phenomena in case of the immune response. And another is the activation of the complement system. And PPRs, they are able to activate the complement system. Ultimately, the microbes will kill. And phagocytosis, they can enhance the phagocytosis with the help of the PPRs, and activation of pro-inflammatory signaling pathway. So these are the very important function of the PPRS and pattern recognition receptors. Pattern recognition receptors, it is divided into the three group. One is the PAMPS. Some PPRs bind to the microbes like mannans, lipopolysaccharide, and peptidoglycans and induce the phagocytosis. So these are the lipopolysaccharides and peptidoglycans and mannan. Mannans means mannose binding molecules which are present on the surface. So it come under to the PAMPS. And another PPRs can also be found into the plasma membrane or into the endosomes and the cytosol, facilitating identification of the intra and extracellular pathogens. So their role is very important, and the host PPRs, they are able to recognize the PAMPS. And another receptor that is known as the LLRS, L-type lectin receptor. Mannose receptor bind to the mannan present on the bacterial, fungal, and parasitic cells. And Adactin 1 bind to the beta glucan present into the fungal cells. And third one is the scavenger receptors. Scavenger receptor A bind to the lipopolysaccharide and lipoteichoic acid which are available on the surface of to the bacteria. Basically the gram positive bacteria are there. So these are the different type of the recognition, pattern recognition receptors are there which are present on the host cell as well as the microbes. And fourth one is the toll-like receptor. And the toll protein was discovered in the Drosophila melanogaster and by Christine, Nucilene, Bolhard and Eric Vestuus in 1985. Then after 10 toll-like receptor have been discovered in human and 13 in mouse. The toll-like receptor are a PPRs and that sense conjugate molecule pattern and can recognize the pathogens like virus and bacteria. The toll-like receptor are transmembrane protein type first having the 3 domain. Domain first it is a LLR. You can see it here is the leucine rich domain are there. So these are the plasma membranes. It is the AR domains. Here is the leucine rich repeats are there. So you can see it is present on the surface of the host cell. They are able to recognize the particular molecules which are present on the pathogens. The LLR is leucine rich repeat motif help in recognition of the pathogens. And second is the TLR are the transmembrane domain, and third one is the cytoplasmic toll like interleukin receptor first, that is known as the TIR domain. It initiates signaling by interacting with the signal transduction adapters. And TLR are found widely on dendritic cells and macrophages and NK cells. It is also found onto the T cell and B cell. TLR is very important receptors are there. They are found into the T cell as well as B cell. Even it is found into the normal cell like to the fibroblast, epithelial and endothelial cells. So upon activation of the TLR, stimulate the antigen induced signal transduction pathway by recruiting adapter protein into the cytosol of immune cells. The adapter protein serve to mediate in other protein interaction and cause the activation of the other downstream proteins like protein kinase. So these are they are able to activate the downstream protein like protein kinases TBK1, II, epsilon. So these are the two protein kinases. They are your TLR. They are able to activate, and these downstream protein further amplify the signal and finally lead to the suppression of upregulation of the gene and carry out to the inflammatory response. So after inflammatory response, the microbes they are not able to enter into the blood stream. So it is a very important phenomena are there that is governed by to the toll-like receptor. And often these events eventually lead to cytokine productions and proliferation of cell enhance the adaptive immunity. So these are the very important phenomena are there. They are able to produce the cytokines and proliferation of cell enhance the adaptive immunity. If the TLR, toll-like receptor identify the factor in bacteria, it is phagocytized and digested and the antigen presented to the CD4 T cells, T cells. And the infected cells may shut down its growth and protein synthesis activity and undergo apoptosis if the viral PAM is identified. So these are the very important things which are available on the surface of the host cell. They are able to recognize the different type of the factors, different type of molecules which are present on the surface, or ultimately they will the immune response will take place. And another thing I want to discuss, it is another component of the immune system, that is known as the NK cells, natural killer cells. The NK cell are larger granule lymphocyte and develop from the lymphoid progenitor cells. That is, you can say it is the lymphoblast. So each and the lymphocyte which are generates by the generate in the bone marrow. The NK cell proliferate and mature in the thymus, spleen, bone marrow, lymph node, and tonsils. So you can see it here is the differentiation. Their site it will be different, like thymus, spleen, bone marrow, lymph node, and tonsils. In human, peripheral blood uh, natural killer cell account 5% to 10% of the lymphocyte. So if you see the actual population of the NK cells, it would be 5 to 10% of total lymphocyte. NK cell are cytotoxic and contain proteins such as perforin and proteases known as the granozymes that help into the cell lysis. Defensin, it is another very important proteins are there, and NK cell secreted antimicrobial directly kill bacteria by disrupting their cell walls. And cytokine like interferon gamma and TNF alpha. So these are the two very important cytokines which are released by the NK cells, and it act on the macrophages and dendritic cell to further enhance the immune response. Uh, Inhibitory receptors signal regulate NK cell killing. How NK cell regulate the killing activity? So you can see it here is with the help of the figures are there. It is a intracellular pathogen. You can see into the red color. Here is the MHC first molecule. Here is the pathogenic antigens, they can recognize it. Here is very important things are there that is known as the here is the cytotoxic cells and MSC first inhibitory receptor which are present on the surface of your host cell. They are not able to recognize it. Here is the MSC first, which are not which is down regulated by the pathogens. Here is MSC first molecule is missing, then your NK cell activate, or ultimately it will kill. The most healthy cell in the body express class first MSC molecule which are recognized as a self. These MSC molecule are recognized by inhibitory receptor. You can see here is the inhibitory receptors are there on the NK cell, which make them inactive and prevent from the killing any healthy cell. So your healthy cell it will not kill, only infected cells and tumor cells. Virus infected cell or tumor cell they will kill. Otherwise your NK cell they will recognize by the particular receptor, or ultimately your healthy cell will not kill. The infected or cancerous cell frequently lost the MSC first molecule, and thus their self tag make them vulnerable to the NK cell killing. Apoptosis via cytolytic granules. So you can see it here is the NK cells are there. They are able to produce the various granozymes, or it will help into the apoptosis. When such molecules are released near to the infected cells, granozymes and perforin are in this cytoplasmic granules. And when rich molecules are released near to infected cell perforins and create plasma membrane pore allowed to granozyme and their enzyme to enter, causing apoptosis. Granozymes are serine proteases stored in the granules and delivered by the perforin and then induce apoptosis. So your cells, your tumor infected cells, it will kill with the help of NK cell. NK cells, they are able to produce the granozymes. And virus infected cell and tumor cell killed by the antibody dependent cytotoxic cells. So you can see it has some virus infected. You can see here is the NK cells are there, here is the antibody receptors, here is the antibody. You can see it is the target are infected with the virus, cells infected with the virus. You can see here is the black tag are there. Then your antibody will be recognize it, or antibody these antibodies it will recognize with the help of the antibody receptors which are available on the NK cells. And the NK cell express CD27 receptor and CD16 bind to the Fc regions, and these anti-tumor antiviral antibodies, and therefore the target cells is destroyed by the NK cells. So these are the mechanism of the NK cell. Ultimately they will remove the virus infected cell and tumor cells with the help of the antibody dependent cytotoxicity. And another very important role of the NK cell, surveillance of the surveillance for the presence of the tumor cell. NK cell constantly come in contact with the many cell and act as a surveillance. So it will provide the surveillance of your our immune systems. NK cell lack antigen specific receptor and even target cell without prior exposure up to the antigen. This is the important as T cell and other immune cell cannot recognize cell in the absence of the antigen. Once NK cell recognize tumor cell, cytokine like receptor necrosis factors and interferon gamma are released to enhance the immune response. So it work as a surveillance systems. If any infected cells are there, they will kill it. If a healthy cell, they will not kill. So the NK cell, they provide this surveillance to our immune systems. And dear learner, I want to summarize my session of the part first. So you have learned the innate immunity is the body first non-specific defense present at to the birth and active before the infection. The different type of physiological barriers, different type of the barriers are there, like physiological phagocytic inflammatory barrier protect innate immunity. And another thing I have discussed, the pattern recognitions receptor. Various pattern recognition receptors are there. They can recognize by the particular host cell and several molecules which are available on the, they will bind or ultimately they will kill it. And third one is the you have learned the toll like receptor initiate immune response by recognized pattern of the pathogens. And NK cell are cytotoxic and other resistant to cancer, virus and graft rejection. Dear learner, we will take a short break. Don't go away. We will come within 2 to 3 minutes.
Do you wish to be part of a television studio like this, or any other media system, or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism New Media Studies. Here in the School of Journalism New Media Studies, we are offering program for all set of people to learn and enhance their media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs are offered through online mode with a sophisticated learning management system. SOGNMS offers programs from PhD in journalism and mass communication till certificate level. We have masters in journalism and mass communication. The eligibility is bachelor's degree, and we are introducing this program in 3 languages: Hindi, Tamil and English. Students from national and international students can join either through online mode as well as in the distance mode. We have 4 postgraduate diploma programs, specialized areas in the field of development communication, digital media, electronic media and journalism and mass communication program. And all these 4 programs are available in distance mode. 2 programs are available in online mode, development communication and digital media. And we have specialized certificate program in community radio. IGNOU employs various media and communication tools for better teaching and learning process. School of Journalism and New Media Studies utilizes this media system for delivering all our programs. Come and join with us for exploring a new career and enhance your media skills.
Dear learner, let me start part 2nd. I have already discussed it. In this part, I will discuss related to the complement system. Complement system is a basically group of the protein. They will organize it, or ultimately they will reduce the infection. They will kill the pathogens. The complement system, the Paul Ehrlich and coined the term the complement to describe the activity of blood serum that complete the action of the antibodies. The complement system is the collection of the soluble and cell bound proteins and produced by the hepatocytes in the livers and gastrointestinal monocytes and macrophages. So here is a they can produce by the hepatocytes in the liver, gastrointestinal monocytes and macrophages. In circulates the serum in inactive from the proenzyme. So basically your protein, the complement protein, it is in active form. After proteolytic cleavage, it will change into the active form of your complement proteins, and thus the complement cascade promote the inflammation and attack pathogens self membrane and help antibodies in the killing of the pathogens. What is the function of the complement system? The complement, they causes the lysis of the cells, pathogen like bacteria and viruses. It promotes the phagocytosis of various particulate antigen via opsonizations. And complement receptor bind to the many cells of the immune system, and this trigger a specific cell function cause the inflammation and subsequently a release of the immune regulatory substances. So, several immune regulatory substances they will release or they will clear the infections. It also help to the clearance of the immune complexes from the body. So, these are the function of the complement system. And there are the three established pathway of the complement activation, namely the classical pathway, lectin pathway, and third one is the alternative pathway. In classical pathway, it began with the formation of the soluble antibodies and the antigen complexes. These antigen present on the surface of the bacterium. So, several antigen which are present on the surface of the bacteria, your your soluble antibodies they will bound it. And it is important to note here that to the C1 macromolecules complex is involved in the initiation step. The C1 macromolecule complexes has the 3 elements and 6 unit. You can see 1, 2, 3, 4, 5, 6 unit up to the CQ, C1Q, and 2 unit up to the C1R, and 2 unit up to the C1S. So, these are the several molecules or they will form to the C1 macromolecule complex. C1 complexes bind to the globular head to at least 2 Fc site up to the antibodies for the stable C1 complexes antibody interaction. So, here is the role of the antibody is very important. Your C1 molecules they will bind to the Fc site of your antibodies. And C1Q, you can see it here is your C1 molecules, they are able to recognize it to the particular antibodies, Fc region of the antibodies, or they will perform the cascade of the reactions, or it will change into the making of the C3 convertase and then C5 convertase, or ultimately they are able to form the MAC or MAC, and then it will recognize other proteins are there, or ultimately they can create a pores or your bacteria will kill. So, I want to discuss one by one in this step. C1Q bind to the Fc portions of to the antibodies that activate C1R and further activate C1S, and C1S cleaves C2 and C4. You can see it here is the C1 and C2, and C2 cleaves into the larger C2A and smaller C2B that diffuse away. Large C4B and smaller C4A are formed. The C4B2A complex called C3 convertase. So, here is the complexes are there. You can see it here is the C4B2A. So, you can say it is the C3 convertase. A single C3 convertase up to 300 C3B molecules and represent a major amplification step, and several C3B molecule combine with the C3 convertase to form the C5 convertase. So, here is the C5 convertase are there, and which hydrolyze C5 and C5A diffuse away and C5B. C5B interact with to the another component to form a membrane attack complexes. So, here is MAC will form. MAC they are able to create a pore in the bacterial cells, and formation of MAC membrane attack complexes, and C4B and C6, C7, C8 and C9 protein interact to form the MAC. You can see it here is the several proteins are there, they are able to combine it, ultimately they will form the MAC. The C5B formed by cleavage by to the C5 convertase and provided an provided an attachment site for to the other component of the MAC. And C5b unstable required to the binding of the C6. You can see it here is the C6 are there for stabilizations of your MAC membrane attack complexes, and further C5 and B6 bind to the C7, and then complexes now has the hydrophobic areas exposed and would allow to insertion of the plasma membrane to the target cells. And C5b67 attached to the C8 and C5b67 and 8 C8 complexes attached to the cells and create a pore around 10 angstrom diameter. But this is just sufficient to lyse the red blood cell, but not to the pathogen. Thus, its involvement of C9 is a very crucial. If complement system is activated, it can kill the pathogen with the help of the C9 factors are there. And about 10 to 17 molecule of the perforin like C9 molecules of the complex capable of insertion of the pathogen membrane. So, you can see it here is the C9 molecules are there, here is the complexes C5, 6, B6, 7, 8 molecules, or here is the poly 9. The poly 9 they are bind together. So, it is a complete membrane attack complexes are there. The complete MAC consists of the C5, B6, 7, C8 complexes and surrounded by to the perforin like poly 9 complexes and kill the bacteria and virus. So, it is a the complement system are there, they are able to kill the pathogens. Another mechanism, the lectin, mannose binding lectin pathway. It is a part of the innate immune system, no need to the formation of antigen antibody complex. So, in the activation of the lectin pathway, there is no need to antigen antibody complexes, formation of antigen antibody complexes. Pathogen like Salmonella, Candida albicans have the mannose residues on their surface. The mannose residues bind to the MBL and activate the pathway. The MBL mannose binding lectins is produced in inflammation as it is the acute phase proteins. The mannose associated serine proteases 1 and 2 bind to the MBL and form to the complex, lead to the cleavage up to the C2 and C4 proteins. And the further step are similar to the classical pathway up to the complement activations. So, you can see in the next slide, here is the lectin pathway, here is MASP 1 and 2 mannose associated serine proteases bind to the MBL and form to the complex, lead to the cleavage of C2 and C4 protein. And the further step are similar to the classical pathway of the complement. So, you can see it here is only difference of the starting, otherwise all the things, all the pathway is the same. Only this place here is the MASP1 and 2 bind to the MBL. So, you can see it here is the mannose binding lectins bind to the foreign surface. So, or ultimately they will activate to the complement system, or they will kill the pathogens or the tumor cells. And third one is the alternative pathway. The alternative pathway does not need to the antigen antibody complex formation for its activation. It is initiated by the molecule presence on the surface of the pathogen. So, several molecules which are present onto the surface cell surface of the pathogens. The alternative pathway can be activated by a cell wall component from the gram positive and gram negative bacteria, and therefore it is a often considered part of the innate immune systems. So, you can see it here is the alternative pathway. Alternative pathway are there for the killing of the pathogens. alternative pathway of the complement system. Here is the serum C3 undergo spontaneous hydrolysis to form C3A and C3B, and C3B bind to the factor B. You can see here is the factor B are there. They will bind to form a complex, and binding of C3B exposed to the site for factor B and allow to binding up to the factor D. Here is the factor D. They will allow to bind with these factors. And factor D is cleave. That C3B bound to the factor B generate C3 convertase. Here is after binding of this factor, it will change into the C3 convertase. And properdine is required to stabilize the C3 convertase, and now C3 cleave rapidly in this act as a major amplification step, and now C5 convertase is generated, and that cleaves C5 to C5A and C5B, and the C5B goes on to the take part up to the MAC. Here is the membrane attack complexes will form. Another thing I want to discuss, it is another component of the immune system, that is known as the phagocytosis. The cell internalize the component by to the particular type of endocytosis called phagocytosis. You can see here is the bacterium, it will internalize by the particular cells, or ultimately they will kill it, they will phagocytize it, and it is not a antigen specific and is part of the innate immunity. Many cells such as to the macrophages, these cells are basically macrophages, immature dendritic cells, and neutrophils are involved into the phagocytosis. Phagocytosis is take place in the macrophages, immature dendritic cell and neutrophil cell, and several receptors help in the phagocytosis of the opsonized pathogens, and there are the several receptors are there, like Fc receptors. In other receptor like toll-like receptor, lectin, scavenger receptor are also crucial into the pathogen uptake. Process of the phagocytosis. So you can see it here is the several steps are there. Ultimately, the particular tumor cells are the virus infected cells, they will phagocytize. The cell surface receptor onto the phagocytes bind to the opsonin, and PMOs represent onto the surface of the pathogen. So these are the PMOs, is nothing, it is a particular molecules which are recognized by to the particular receptor which are available on the host. The phagocytosis is triggered due to the receptor clustering. The phagosome eventually pinched off, and the cell membrane envelop the pathogens. And the phagosomes is an acidified body forms, and due to the eventual fusion of the phagosomes are the late endosomes. The phagosomes is degradation of to the target cell occur due to the hydrolysis of the hydrolytic enzymes. Another thing is inflammation. Inflammatory response is the cascade of the event that is triggered by to the damaged tissues, tumor or pathogen invasions. So you can see it here is the several sign of the inflammations. Here is you can see here the 4 signs of the inflammations are there. It is a part of the innate immune response, and it is not a pathogen specific. If any pathogen will come, inflammation will take place. It is a protective response of the bodies and involved into the several element like immune cells, blood vessels, and various molecular mediators. It reduces the infection, remove to the damaged tissues, and also initiate to the healing process. The type of inflammation, basically there are the 2 type of inflammation. One is the acute inflammations. It's short-term process and triggered immediately after infection, and cause the acute inflammation include the pathogen, toxin, allergen, and allergens and burns. Microbial pathogen are recognized by to the TLR, toll-like receptor. Here is role of the toll-like receptor is a very important. If it not recognized, the inflammation will not take place. Neutrophils, macrophages migrate to the site of inflammation under the influence of the cytokines and chemokines. It is activated by the pathogen associated molecular patterns and damage associated molecular patterns. So, there is very important molecular patterns are there, PAMPS and DAMPS. Then inflammation will take place. Second is the chronic inflammation. It is prolonged and last of the month of to the year. Macrophage, plasma cell and lymphocytes are involved in the chronic inflammation, and several diseases like allergy, cardiovascular disease, diabetes and chronic obstructive pulmonary disease are caused by the chronic inflammation. It is also associated with the condition like hay fever and atherosclerosis. Event of the inflammation process. There are the several events which are involved in the inflammation process. One is the vasodilation. This involved into the increase of the diameters and permeability up to the blood vessels that allow more blood flow to the site of damage or injuries. The readiness of tissues increasing the temperature at to the site of inflammation and begin to the arteries and to the eventually lead to the enlarging the capillaries. The plasma moves due to due to the increase of permeability up to the blood vessels and resulting into the larger concentration of cell and that enlarge to the blood vessels. The vasodilation is a very important event of the inflammations. And another the increase into the capillary permeability, it is another very important mechanism of the inflammations, and this help to the move to the fluid from enlarged capillary into the tissue space. There is a fluid accumulation that cause the oedema. Influx of the phagocytosis, it is another mechanism of the inflammations. Influx of phagocytosis occur to the inflammation site due to the increased permeability of the capillaries. So, the number of the cell they will move, but pathogens they are not able to move at a particular site. So, it is a very important component of the immune systems. And another thing I want to discuss, the degranulations of the mast cell. Here is the, you can see in this figure, it is a mast cell bit to the IgE, mast cell bit to the IgGE antibody attach. Here is the antigens, antigenic stimulation are there, antigenic come bit attach, antigen come and attach to the IgE, and various chemical mediator are released like histamines, trypticase, kinase and various cytokines and chemokines, ultimately degranulation of the mast cell. So, you can see it here is the degranulation of the mast cell. It is governed by to the particular cytokines and histamines and various chemical modulators are there. Dear learner, I want to summarize part second. You have learned the component system, complement system serum, protein help antibody and other cell killing pathogens. So, I have already discussed it. There are several pathways are there of the complement system. You have seen to the classical alternative lectin pathway, or each pathway activate differently, and wound tissues damage, pathogen invasion cause inflammation, cell vessels and molecular mediator are involved. The allergies, transplant and autoimmune diseases cause the chronic inflammations, and mast cell degranulation and macrophage activation release several inflammatory mediator. Dear learner, you have learned the various things related to this topic. In this session, if you want to do the detailed study, you can go through the IGNOU SLM BB BZYET141. It is elective courses. You can go through, you can clarify each and everything related to the this topic complement system first. Thank you.
Are you a fresh graduate? Do you want to fulfill your educational aspirations?